talking about the heartbeat today. We're going to start with what could be a review for some of you, what actually drives the action potential within our pacemaker cells, as well as the rest of the myocytes that give us this actual contractile response that we're looking for within the heart. We'll talk a little bit about calcium handling. And we're going to talk about the signaling pathways that drive this heart rate response. That's important because you need to know that to understand how does parasympathetic and sympathetic activation alter your heart rate, which is going to be important both to those interested in strict exercise responses as well as those interested in pathology. And then we're going to finish with putting that all together with atrial fibrillation, which is a very common arrhythmia that we see in the clinic. So action potential is the electrical stimulus created by a sequence of ion fluxes through specialized channels in the membrane. So a lot of times we think of this sarcolemma of the cardiomyocytes that leads to cardiac contraction. And if we actually look at over here on the right, here's our voltage as a function of time. We're in milliseconds. We've got some resting potential down here. It's usually around -70. And we've got whatever it may be. At this point, just think of it as a stimulus that's going to drive this membrane potential. It's going to depolarize it a little bit here. And there we can see here some failed initiations. Now, there was a stimulus, but it wasn't a strong enough one. If it gets above a given threshold level, we then get full depolarization. We get the full action potential, followed by repolarization, a refractory period, and a resting state. So this is what we're really dealing with here, what's causing this response within cardiac myocytes. Let's just do a little highlighting here. All right, so again, some of this is a little bit of a review if you've had AMP or 603, but we're going to go through it in a little bit of detail here. So the SA node, the sinoatrial node, primary pacemaker site within the heart, and we'll talk about it in isolation here to begin with. Characterized as having no true resting potential, and it generates a regular, spontaneous action potential. And it has three phases that we're really interested in here in the class: the phase 4, phase 0, and phase 3. We'll talk about each of these in a little detail, but I'll just give you kind of the initial definition. So phase 4 is the spontaneous depolarization or pacemaker potential that triggers the action potential once the membrane reaches the threshold. I'm going to slide down here a little bit and show you this. So we've got millivolts here as a function of time. Here's that threshold. Here's phase 4, spontaneous depolarization. It triggers the full action potential once it reaches that threshold. Phase 0 is the depolarization phase of the action potential. So we get this, here's phase 4 that brings it up, phase 0 that is the full action potential, and then phase 3 is repolarization. Unless I tell you specifically and highlight it, you do not need to know that between -40 and -30 millivolts, or repolarization at about -60. There's a couple places where that does matter, but if I don't tell you to know it, don't worry about these specific numbers here. They're kind of just there to help fill in some gaps. So these are the three main phases that we're interested in here within our SA node. And this just continues. We can see here after repolarization, we start phase 4 again. There's no resting. It bottoms out, and this goes over and over. And this is at the SA node, that pacemaker cell, or grouping of cells. So what's driving this action potential is the movement of ions. And it's principally calcium, potassium, and sodium across the membrane. And they're doing this through ion channels that are going to open and close and become inactivated throughout this cycle. And it's this sequence of ion channels opening and closing, the signaling of these ions, that give us the action potential we need. And what we'll see pretty quickly is if you alter that, you then alter the heart rate. And if you screw with it, that's where you start getting the arrhythmia. And small screw-ups really have a pretty profound impact, and that's where we'll finish things off today. So principally, within the SA node, calcium, potassium, and sodium. And really, it's just a, again, going back to the opening and closing. So here's millivolts, function of time. We open a channel, the ion moves, that ion's got a charge, and therefore we get changes in the membrane potential. That's really all that's happening. The main thing that we're focused on here in this class is what channels are opening and when are they opening? What is the sequence of events taking place? So ions flow, generate electrical current, change the membrane potential. Closure of the ion channels causes that ion conductance to decrease, and something else kind of comes into play. And that lets us fine-tune this response. So what are we already mentioned that calcium, sodium, potassium are players here. Let's see what events are transpiring at the SA node to give us this action potential. And we'll start with the image here. So again, millivolts as a function of time. Here's this threshold that I mentioned in that very first image that we've got to get above to get that full action potential. And we've got calcium, inward rectifying calcium, there's T-type and L-type, and there's these funny channels. And then I've got down here the external and the internal side of the cell and the directional movement of these various ions. So I'll kind of go back and forth here. This is just the image. I'll highlight what's really important kind of in these bottom portions. So 3 ions, important at the SA node. They give us this pacemaker action. So at the end of repolarization, when the membrane potential is very negative, so it's down in the negative 60, so we're talking down in this range here. The ion channels open that conduct slow, inward depolarizing sodium currents. And these are referred to as funny currents. These funny currents is what gets us going back up the hill. So down here at this very bottom, we can look at it from here, reaches its very bottom, that would be here as well. These funny channels, abbreviated IF, allow sodium into the cell and let this membrane potential slowly work its way up. We can see that, as we get around here about this portion here, we start seeing some calcium channels open. So as the membrane potential reaches about negative 50, so this is one where the negative 50 matters. Here and the next one are the only places where this really makes a difference. As the membrane potential reaches about negative 60, the T-type calcium channels open. When it gets a little bit more depolarized, so it gets a little bit closer to 0, so at about negative 40, the long-lasting L-type calcium channels open. So we've really got a lot of calcium coming into the cell at this point. It is this L-type calcium channel opening that is the primary driver or primary cause of the phase 0 that gives us the actual action potential that's going to initiate the heart rate or the heartbeat. Again, this is all at the SA node. So I'm going to slide up here and just review what we have. So funny channels started up the hill. The T-type calciums push it a little bit more. The L-type calcium takes it over the edge and gives us the full phase 0 depolarization. We can see here, once we get to the top, this inward rectifying potassium channel opens and gets things back to normal. So repolarization, phase 3, potassium channels open. There we go. And at the same time, This is important because we'll bring this up when we start talking about Afib. The L-type calcium channels become inactivated and close. It's not just about turning on at the right time, it's also about turning off at the right time. Very coordinated sequence of events. So this is at the SA node. The action potential that starts at the SA node then disassociates throughout the atria, gets to the AV node, that goes through and gets ventricular depolarization and ventricular contraction. So everything's really starting here, and it's going to then set the rate of our heartbeats to the heart rate. We know that there are parasympathetic and sympathetic influences. So I'll give you a second here, let you write these, and then I'll walk you through what's happening. So one of the cornerstones of exercise physiology is that we can control heart rate through the parasympathetic and sympathetic innervation of the heart. Parasympathetic neurons, they innervate at the SA node, and we know from previous classes that if we've got increases in parasympathetic nerve activity, we're going to have a decrease in heart rate. The way that this is happening is we've got postganglionic neurons release acetylcholine, which bind to muscarinic cholinergic receptors. Turns out, muscarinic cholinergic receptors is a G protein coupled receptor. One way that it's lowering heart rate is it opens potassium channels through a stimulatory G protein and closes the funny channels and the T-type calcium channels through inhibitory G proteins. So here's our acetylcholine, here's our cholinergic receptor, and then our two G proteins. Well, on the stimulatory side, we're getting this opening of the potassium channels, and it's closing or promoting the closure of things like the T-type calcium. If we slide up, you don't need to. If we slide up here, well, if we don't let these channels open, so we prevent the T-type and the funny channels from opening, that's going to make it a lot harder to get back up here to this threshold. If we keep these potassium channels open, this potassium wants to drive this back down. All of this in concert is keeping that membrane potential down. It's suppressing the SA node and what it wants to do, therefore suppresses heart rate. So we can give various drugs or whatever it may be that can bind to these cholinergic receptors and drive this response. When we have this, it refers to as hyperpolarization, and a decreased frequency of the action potential, and therefore decreases the heart rate. Sympathetic neurons also innervate the SA node. and are designed to increase the frequency of our action potentials and therefore increase the heart rate. So, we get postganglionic release of norepinephrine, which binds to beta-1 receptor on the SA nodal cells, and this binding to our beta receptor, we get a G protein, in this case it's a GS, activates cyclic AMP. So here's our norepinephrine, here's our beta receptor, and we get cyclic AMP. We've had this already on the exam, so I'm not going to go into detail, but we've got our G protein, here's our adenylate cyclase, it gives us cyclic AMP. We talked about this pathway. Cyclic AMP augments the opening of funny channels and T-type calcium channels, which therefore increases that initial slope, so it's allowing that membrane potential to get to that threshold faster. This faster increased frequency of action potentials, therefore increases the heart rate. So we've got cyclic AMP through some protein kinases, promotes the opening of these funny channels. So cell signaling is not always, when we're talking about responses and adaptations to endurance exercise and strength training and those sorts of things, it's going on and altering gene expression, give us that adaptation. But it also is pretty innately involved with the adjustments to exercise. The rapid responses that we would see here, we can really give someone a beta blocker, we give beta blockers clinically, lower someone's heart rate, reduce the work of the heart, because the work of the heart, is typically calculated as your heart rate times your systolic blood pressure. Well, if I can give a beta-1 blocker, that's going to competitively inhibit these beta-1 receptor and prevent norepinephrine from binding, and therefore it's going to we're not going to get this response. It's not, despite having high norepi levels, these funny channels and T-type calcium channels are going to stay down, and therefore we don't have the increased heart rate, therefore we don't have the increased work of the heart. That's an important thing if you've got a cardiac patient where you want to keep myocardial VO2 down because they've got atherosclerotic plaque, or they're at risk, and those sorts of things. So that's where some of this signaling pathway can really has some importance. We can look at these kind of the normal sympathetic and parasympathetic stimulation. So these again are action potentials from the SA node. In green here is the normal frequency. It's not going to be on this figure won't be on the exam. So we can see we got this slope where our funny channels and our T-type calcium channels are driving it up. We get our action potential and it comes back down. If I have parasympathetic, I'm preventing the opening of the funny channels. The slope, it takes a lot longer to get up to this threshold. So because it's taken longer to get here, it takes longer to get that action potential and therefore heart rate. If I steepen that slope because I'm promoting the opening of those channels, I can have that action potential very quickly. So that's in the SA node. It's just the pacemaker. It's driving the rate. Now what's the action potential within the cardiomyocytes? These would be the cells within the atria and the ventricle that are actually going to have some of the contractile properties that we need. And we need this action potential to drive that response. Action potential in the typical cardiomyocyte is composed of 5 phases, 0 through 4, beginning and ending typically with phase 4. We're going to start with phase 0 because that's just where I like to start. So phase 0 is depolarization. and I'll kind of walk you through the key steps here. There's a little bit more information here than you necessarily need for the exam, but I think it's important to kind of help fill in some of the gaps. So phase 0, depolarization. So let's look at this figure here. We've got time on the x-axis, millivolts on the y-axis. Here's phase 0. It is this very rapid depolarization. Here we get sodium currents, rapidly depolarize the membrane, and we get some L-type channels open and cause an influx of calcium down its concentration gradient. So we can see here in our image, it's sodium's really driving this, and we get a little bit of calcium kind of towards the very end of the phase 0. It's not perfectly lined up, but you can start to see what's happening. We have a little bit of early repolarization, so that's this little drop. So coming back down is repolarization, so it drops down, and that is through some potassium channels. During this plateau phase, so we've got phase 1 dropping down, phase 2 is this plateau phase. Some L-type channels are still open, and some are closed, and there's this small inward current of calcium. And we get some leaking of potassium. It's on the next page, but these two are kind of electrically balanced, so they kind of cancel each other out. We can see here this kind of balance. We've got sodium going out very initially, sodium going in, calcium going in. Then the calcium starts to waver off. Potassium's very little at the beginning, starts becoming stronger, and that's kind of driving this phase 2 and phase 3 responses, this interaction between these. And this early balance between them is what gives us kind of this plateau. I just said that there. Phase 3 is repolarization. So during this repolarization, when that slope is going down, calcium channels are gradually inactivated. So basically, we're turning these calcium channels off, preventing that movement of calcium. We've got a much more persistent outflow of potassium that's exceeding the calcium, so that's going to drive that membrane potential back down. That's that repolarization phase. So closing the calcium, persistent flow of the potassium that exceeds calcium brings that membrane potential back towards resting. Notice I didn't highlight the -90. You don't need to know that. It's back towards resting. And that gets it set for starting the whole process over again. Phase 4 would just be keeping things at resting membrane potential. I'm not really worried about that at this point. And that's this really phase 4 here. Sarcolemmal sodium calcium exchanger, calcium ATPases, sodium potassium ATPases all kind of keep things down there at the normal. So it's this interaction. We've got to have these turn on and turn off all at the correct time to give us this profile we need. And this is the action potential that then elicits the mechanical contraction. Okay, so if you've had muscle fizz or 335 and even AMP, you know that calcium and the handling of calcium is going to be required in order to get us that force production. And in cardiac muscle, the force of the contraction is highly dependent on peak intracellular calcium concentrations during systole. The sarcomere length, so that's length tension relationship. The myofilament's responsiveness to calcium is also going to come into play. So if we screw up handling of calcium, things just go to hell. We get contractile dysfunction, we start seeing arrhythmias, and these are really what's driving a lot of the pathophysiological conditions we see in cardiac tissue. Okay, so during this action potential within our cardiomyocytes, calcium ions enter via our voltage-gated calcium channels. So this is just an inward calcium current. Two types of voltage-dependent channels we've already discussed. These are the T-type and L-type. The T-type, we've got them. They help with the action potential, but they're really negligible when it comes to really the calcium response here with the contraction. The L-type, they're primarily located at the sarcoplasmic reticulum junction, where the ryanodine receptors exist and can really help trigger this response. So I'm going to slide down here and just I'm going to zoom in a little bit. Okay, so we've got our T-tubule, we've got sodium channels, we've got potassium channels, we've got sodium-calcium exchangers, and down here is our L-type calcium channel. So we just saw what that does above. It helps drive the action potential. Here's our ryanodine receptor, here's our sarcoplasmic reticulum. And just like in skeletal muscle fizz, we can get sarcoplasmic release of calcium that is then going to drive excitation, contraction, contraction. So down here at the sarcomere. So that's a main important with that. Sodium calcium exchanger contributes a little bit, so we get here's our sodium calcium exchanger. But as I've already mentioned, that the main effect is to stimulate calcium release from the intracellular pool within the SR. And this would be our calcium-induced calcium release. The L-type sodium calcium exchangers are going to really help with this SR release of calcium. Another common drug that if you look at cardiac patients that is prescribed are calcium channel blockers. These are used to treat hypertension, angina, certain arrhythmias, and they bind primarily to the L-type calcium channels, because if you can block this channel, not only are you going to maybe potentially alter the action potential and overall heart rate, what are you doing there? It's going to help limit maybe the release of calcium from the SR and therefore reduce some of the issues. And we'll talk about calcium release. It's a main player when it comes to atrial fib. Okay, well, if you've had 335, you know that the sympathetic nerves not only innervate the SA node, but they actually innervate the myocardium and can have a direct impact on the cardiomyocytes. And they have their effect through calcium release, which therefore alters ventricular contractility. That's going to help increase our ejection fraction, which is going to improve our stroke volume, cardiac output, and so on and so forth. So as before, sympathetic nerves release norepinephrine, binds to beta-1 adrenergic receptors. These activate adenylate cyclase. They give us cyclic AMP. In the ventricular myocyte, this cyclic AMP goes on to have multiple effects that all drive changes in cardiac function. So I'm going to highlight them here and go back and forth between our images. So first is phosphorylation of calcium channels causes the calcium channels to remain open longer during the action potential. More calcium, increased force of contraction. So again, I'm going to slide up and down here. So norepinephrine, here's our beta-1 receptor, here's our G protein. That G protein activates adenylate cyclase, gives us cyclic AMP. And first step, L-type calcium channel is going to help keep that open. So we get influx of calcium into, this is, again, the ventricular myocyte, not the SA node. It's going to phosphorylate or promote the phosphorylation of proteins at the sarcoplasmic reticulum, which is going to help enhance the release of calcium from the SR. So that's point 2 here. So here's our SR. It's going to help this process in calcium release. It's going to increase the rate of myosin ATPase, which increases the speed of cross-bridge cycling. And phosphorylation of calcium pumps in the SR to increase the speed of calcium reuptake, which is going to improve the rate of relaxation. This is an important thing with cardiac patients. It's not only just about systole, but diastole comes into play as well. Heart failure with preserved ejection fraction, which is largely undiagnosed, deals with this rate of relaxation. So this is the signaling pathway in which norepinephrine binds to the beta receptor through adenylate cyclase, cyclic AMP, protein kinases, has this impact on the calcium channels, the SR release, calcium uptake at the SR, and the cross-bridge cycling. Since we're talking about calcium handling, and just mention a little bit about the reuptake and relaxation and filling of the heart. So in order for this to take place, we've got to get this intracellular calcium that we've really focused on thus far. It's got to decline. 4 different potential pathways or methods in which we can reduce intracellular calcium. This is the SR calcium ATPases, so the SERCAs, sarcolemma exchangers and calcium ATPases, and mitochondrial calcium immunoporters. As you can probably guess from previous classes, the SERCAs, so the SR calcium ATPases, they're the main driver. We're talking 70 to 75% of the activator calcium from the cytosol gets taken up through this thing. The sodium calcium exchanger picks up the remaining 25 to 30% with a very small amount through the sarcolemma calcium ATPs and mitochondrial immunoporters. We can just look at that visually here. So here's our T tubule. Here's the mitochondria. So we've got our SERCAs. Here's the ryanodine receptors and the SR, about 75% here. about 25% for this calcium-sodium transporter, and about 1% to like the mitochondria. And then this also gives you an idea about where this is coming from. Also on release, about 75% from the SR and the L-type, about 25% from other sources. In heart failure, the expression of these circas, so right here, is normally reduced and does contribute to the decline in intracellular calcium and diastole. This results in a reduced calcium content within the SR, limiting the amount that it can be released, and therefore appears to be a central cause of systolic dysfunction within heart failure, or at least HFrEF, which is heart failure with reduced ejection fraction. Okay, so that's kind of normal function with a little bit of pathophysiology. We're going to finish up here with atrial fibrillation. It's the most prevalent cardiac arrhythmia in the developed world, about 6 million people in the U.S., and it's likely to increase or double by 2030 because of the aging population, because of comorbidities, and we see it with heart failure quite a bit. It's actually a concern NASA has because there's changes within the atria with spaceflight. So it's a pretty important type of arrhythmia for us to look at. Because of that, we know a little bit more about it than we do in some of the others. So I either write out atrial fib or I just have it as AF here. So AF, or atrial fibrillation, is a supraventricular tachyarrhythmia characterized by uncoordinated atrial activation with consequent deterioration of atrial mechanical function. So we're not talking about the ECG here and what we're going to really see, but I do want to give that put that into some practice, so we'll look at it in just a second. Or just a second. But on the ECG, atrial fibrillation is characterized by the replacement of that consistent P wave. So if you know much about ECGs, actually, I can show that down here. So here's our SA node. We get dissociation through normal electrical pathways to the AV node, and we get contraction. And therefore, we get this nice little P wave, QRS complex, and T wave, and it's nice and regular. With AFib, we're replacing this constant P wave by rapid oscillations or fibrillatory waves that vary in amplitude, shape, timing. They're irregular. They can be pretty rapid, and they're all over. So we get these abnormal electrical pathways. So not only do we have our SA node, but we've got things taking place out here, little ectopic sites within the atria that are also firing, and therefore things are just all over. So now you see something that looks a little bit like this. P waves all over, very irregular. This creates a problem. You think, oh, well, it's just the atria. It's not like it's pumping the systemic circulation. Do we really care? And the problem is that over time, it can really create some issues. So here things like atrial stretch, fibrosis. We're going to talk primarily about altered calcium handling. That's really what's at the root of the matter here. All of these things give us AFib. Okay, so what happens if you screw up the atrium? Well, you don't get that atrial kick, which helps fill the left ventricle, and it's only about 15 to 20%, but it still matters. So loss of the atrial kick, you've got irregular rhythm. Well, that might screw up coronary flow a little bit because coronary flow only occurs during relaxation. If you don't get good relaxation, then you're not getting good coronary flow. And in some cases, it can screw up ventricular rate. That gives us LV dysfunction, which is then going to give us symptomatic cardiovascular issues. So it is an important thing for us to deal with. Just to give you a little bit of more background, we can classify it in a variety of ways. Paroxysmal, persistent, and permanent, and that's just based off the duration in which you have the symptoms or you have the Afib response. There is remodeling, and we could go in over this for days, but we're not going to do that in this particular class. This is where I get to plug my class for the fall. So in the fall, we'll go over this in greater detail in 605, which is Pathophysiology. It's a new class, so we'll talk about these in greater detail. So it'll be a little bit less of the signaling and more of the more clinical pathophysiology associated with these. But we've got these three different types. So each atrial fibrillation, and they're happening all the time, all over, requires some initiation by a trigger acting on a vulnerable substrate within the myocyte. And we can have comorbidities, things like age is going to increase or promote the vulnerable substrate for initiation. Things like hypertension, heart failure, cardiac valve disease, those are some of the classic ones. We're starting to see it with age, and that's where we're thinking some of the prevalence is going to increase over time. There is genetic predisposition for Afib, so we're not going to get in kind of the genetic side here. We're going to focus more on what we see clinically here with some of these comorbidities. I'll let you guys write these a minute and I'll talk about them. So one of the primary mechanisms for Afib are these focal apoptotic or triggered activity. And they're caused usually by something referred to as early after depolarizations, or EADs, or delayed after depolarizations, DADs. EADs are favored by delayed repolarization. So I'll talk about these down here and then kind of scroll back up and highlight. So here's early after depolarization. This dotted line here, this is our normal action potential. We just went through the steps that drives this phase 0, 1, 2, and 3. Early after depolarizations occur as if this plateau lasts a little bit too long. We get, instead of coming back down here in repolarization, we get this early after depolarization. So we get this up like so. And it can consistently go up like that, or it can be just a little spike. So that's early after depolarization. And it's favored by this delayed repolarization. The longer this plateau lasts, the more likely we're going to get some of these little EADs. The DADs, or the delayed after repolarizations, occur if we screw up the way we're handling the calcium within the myocyte. So we have a normal action potential, but so again, we're at the myocyte here. We're not at the SA node. It should not trigger itself. It should be waiting for a signal. But in a DAD, everything comes back to normal, but then it gets this little spike. And if that spike is enough, it can trigger its own full response. So this early after depolarization is due to AP depolarization prolongation, and this is usually due to abnormal SR calcium release. Let's look at it in just a little bit more detail. So the early after depolarizations, we know what drives the normal action potential within a cardiomyocyte. We can start seeing where maybe some of this dysfunction that's driving this Afib comes into play. So as mentioned, the EADs, occur in a setting of a prolonged action potential. So instead of that action potential going up and a little bit of plateau and then coming back down, we've stretched that out. And if you remember, during a normal action potential, the L-type channels, calcium channels, they open, but they also, we highlighted it, have to become inactivated. So they turn off, they close, become inactive. If we get reduced potassium repolarization currents or increased depolarization, and it prolongs this action potential, so things come up and then things are a little bit slow to go down, this prolongation allows time for those calcium channels to become inactive and then flip back on. It's like having a light switch on, on a timer. You've flipped it off, but it has the potential to flip back on. If we've kept up here, because we've delayed this action potential, it allows time for the L-type calcium channels to recover from inactivating, and then thus allowing them to become active again, results in an influx of calcium again, and causes that little spike. They're supposed to be turned off and let everything go back to normal, but they kick things back on. So if we can look at this visually here, our sodium, so here's, for instance, our sodium channels. Here they are. Sodium, our potassium channels. So we've got a little bit too much sodium. We've got less potassium. So less potassium, they're a little bit inactive. The sodiums are a little bit active, and this increases the action potential duration. That increased action potential duration lets us this point C. Allows this L-type to be a little bit more active, to turn itself off and flip back on. And that little spike there, this turning back on of these L-type calcium channels, is what gives us this spike. If we've re-spiked it, we've told that myocyte to contract again. So it's contracted, and instead of fully relaxing, it's contracting again. And now if we've got all these cardiomyocytes all over the atria doing this, it's not this nice coordinated action. We've got firing all over the place. We don't have nice synchronous activation. The DADs primarily arise from dysfunction of iodine receptors because of things like enhanced calcium calmodulin dependent protein kinases. So if we look here, this is our normal action potential, spike, repolarization, a little bit of plateau and back down, and here's our DAD. We get this increase in CAM kinase 2. That's going to increase phosphorylation of our iodine receptor, and therefore it's going to, we're going to get a little bit of calcium out. Let's see, do I want to have you highlight that? I'm just going to have you highlight point A. Point B I'm going to go over, but this is the main one. So it also can increase SR calcium load by increases in the uptake. So it's going to help intake calcium so that when it dumps even more. And there is some diastolic release from the SR. So in diastole, a little bit of calcium basically is leaving from the SR and therefore can trigger an additional action potential. So if it reaches, in this case, this little bump, it wasn't much, it didn't give us a full response, but if it is enough, it can trigger its own action potential. Again, this is the cardiomyocyte. It's not supposed to do this unless it gets a signal from the SA node. In these two situations, it's doing it on its own. Because of mishandling of calcium or the mishandling of these channels, this lone myocyte out in the middle of the left atrium fires. And if it fires, it might fire some of the ones around it, and therefore things are out of sync. And the last portion, heart failure. Reason we see AFib a lot with heart failure is because there are so many things going on. First is we get an increase in oxygen stress. Talked a little bit about that in previous lectures. We get loss of calcium homeostasis. Just mentioned that a little bit with screwed up circuit channels. It also has screwed up activation of that calcium calmodulin independent protein kinase 2, the CAM2. Therefore, all of these things, so these three things are all screwed up with someone who has heart failure. Well, the ROS screws up CAM kinase and the calcium handling screws up CAM kinase. We've just mentioned here that if we screw up CAM kinase, we screw up this and potentially get more of these DADs. We find that this CAM kinase can be upregulated through reactive oxygen species and poor calcium handling and exerts a pro effect through multiple actions. Phosphorylate the calcium channels and increase its open probability, so may get more early depolarizations. I'll highlight those two. It can favor phosphorylation of the rhyodine receptor to increase SR calcium leak and cause some of the DADs. Both of which are going to increase AFib, and you've now got someone who's not only got a screwed up left ventricle just because they have heart failure, but now you've screwed up the the left and right atriums, and you're just adding insult to injury. Questions? All right.